Hello. In today's video, we shall learn in detail about chromosomal theory of inheritance. By the end of this video, you will have a clear understanding about different types of chromosomes, linkage, crossing over, and sex-linked inheritance. Let us start with the introduction to genetics. Genetics is a branch of biology that deals with the study of heredity and variation of inherited characters in organisms. Heredity is the biological process responsible for passing on physical traits from one generation to another. The characters or structural differences which provide individuality to a species are said to cause variation among the species. Let us first understand the chromosomal theory of inheritance. This theory was put forth by Sutton and Bowery in 1904. It is a theory of genetics which identifies chromosomes as the carrier of genetic material. It states that gametes carry all the hereditary characters and act as a link between parents and offsprings. Chromosomes present in the nucleus of gametes carry all the hereditary characters. Chromosomes are linear structures with genes located at specific sites called loci along them. They are found in pairs in somatic or diploid cells. During gamete formation, homologous chromosomes pair and then segregate at meiosis. Each gamete contains only one chromosome of a pair. During fertilization, the male and female gametes fuse to form a zygote retaining diploid number of chromosomes. Next are the chromosomes. Chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Each chromosome is made up of protein and a single molecule of DNA. Chromosomes are capable of self-replication and play an important role in heredity, mutation, variation, and evolution of species. Chromosomes which are morphologically and genetically identical are called homologous chromosomes. Presence of whole set of chromosomes in individuals is called Euploidy. It includes haploid, one set of chromosomes, diploid, two sets of chromosomes, etc. The haploid set of chromosomes is also known as the genome. In interphase, chromosomes are in the form of chromatin network. In metaphase, they show maximum condensation and appear short and thick. A metaphasic chromosome consists of two sister chromatids joined at the centromere. The centromere is also called the primary constriction. Besides the primary constriction, the chromosome possess secondary constrictions. Some of the secondary constrictions are called nucleolar organizers because they play a vital role in the formation of nucleolus. Sometimes the chromosomes bear round or knob-like appendages beyond the nucleolar organizers called satellite. The tip of the chromosome is called telomere. Coiled filament in the chromosome is known as chromonema. The thick bead-like structures of the chromonema are known as chromomeres. By the way, if you are getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us look into the different types of chromosomes. The position of centromere vary from chromosome to chromosome, and accordingly, the chromosomes are classified into the following types. First, metacentric chromosome. Centromere lies in the center of the chromosomes, forming two equal arms. It appears V-shaped during anaphase. Second, sub-metacentric chromosome. Chromosomes have arms of unequal length as the centromere lies near the center. It appears L-shaped in anaphase. Third, acrocentric chromosome. Centromere is situated near one end of the chromosome. One arm of the chromosome is very short. Other arm is long. It appears J-shaped in anaphase. Finally, telocentric chromosome. Centromere is present at the very end of the chromosome. It shows only one arm and appears rod-shaped. Next are the sex chromosomes. In humans, X and Y chromosomes are the sex chromosomes. They determine the biological sex of an individual. Female inherit an X chromosome from the father for a XX genotype. Males inherit a Y chromosome from the father for a XY genotype. The human genome is organized into 23 pairs of chromosomes, 22 pairs of autosomes and 1 pair of sex chromosomes. Let us learn about the differences between X chromosome and Y chromosome. X chromosome is longer than Y chromosomes. Y chromosome is shorter than X chromosome. X chromosome contains large amount of euchromatin and small amount of heterochromatin, while Y chromosome contains small amount of euchromatin and large amount of heterochromatin. In X chromosome, the euchromatin contains large amount of DNA or genes and hence genetically more active. In Y chromosome, there is less amount of DNA and hence genetically less active. X chromosomes contain over 1,000 genes. Y chromosomes contain nearly 78 genes. Genes present on X chromosome show criss-cross inheritance, that is, the inheritance of characters from the father to his grandson through his daughter. Y chromosome show linear inheritance. X chromosome found in both genders, while Y chromosomes are found only in males. Next, we shall take up linkage. Genetic linkage is an exception to Mendel's law of independent assortment. Linkage is the tendency of genes to stay together in a chromosome during meiosis of sexual reproduction. Linkage occurs when two genes are closer to each other on the same chromosome. It is due to linkage that the parental traits are seen in the offspring. Linkage reduces the probability of variation through sexual reproduction. The intensity of linkage between two genes is inversely related to the distance between them in the chromosome. Linkage is broken down by the process of crossing over occurring during meiosis. Before we look into the different types of linkage, let us understand the term crossing over in the field of genetics. Crossing over is a phenomenon through which genetic information is exchanged in the germline. Through crossing over, the genes between homologous pairs separate and segregate into different gametes. Crossing over occurs when two genes are located far apart on the same chromosome. Crossing over takes place during the prophase of meiosis I. Next, we come to the kinds of linkage. Complete linkage. The completely linked genes are so closely located in the chromosome that they do not separate and inherit together. This phenomenon is known as complete linkage. Example, the genes for bent wing and shaven bristles of the fourth chromosome mutant of Drosophila melanogaster exhibit complete linkage. Incomplete linkage. The incompletely linked genes are located far away on the chromosomes and can easily separate by crossing over. This phenomenon is known as incomplete linkage. Morgan, while working on Drosophila, found that when genes are grouped on the same chromosome, some genes were very tightly linked while others were loosely linked. The former showed low recombination while the latter showed high recombination. Example, the genes for white-eyed and yellow body in Drosophila were tightly linked and showed 1.3% recombination and 98.7% parental combination. Another example, incomplete linkage in linked genes for body color and wing shape is seen in Drosophila. Parental combination, gray long and black vestigial 83%, and new combination gray vestigial and black long 17%. Next, we shall look into sex-linked inheritance. The phenomenon of inheritance of sex-linked genes is called sex-linked inheritance. They are of two types, X-linked and Y-linked. X-linked or sex-linked genes. Most sex-linked traits are recessively inherited so that heterozygous female generally do not display the trait. In females, two recessive sex-linked genes are required for the expression of sex-linked trait. In males, single recessive gene can express itself. Examples of sex-linked diseases are hemophilia, color blindness, night blindness, etc. Y-linked or Holandric genes, they are expressed only in males. Example, hypertrichosis, that is, excessive growth of hair on pinna of ear. The inheritance of characters from the father to his grandson through his daughter is called criss-cross inheritance. Let us understand criss-cross inheritance with the following example. A color-blind person cannot distinguish between red and green colors. If a color-blind male marries a female with normal vision, then, as shown in the figure, the first generation of offsprings will have normal vision. The daughters act as carriers of the disease. Now, if a carrier female marries a normal male, all daughters are normal or carriers, but half the number of sons are normal and other sons are color blind. Color blindness is a good example of criss-cross inheritance. So today we have learnt about chromosomal theory of inheritance, linkage, crossing over, and sex-linked inheritance. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.